Hey guys, we're going to be talking about genetics part 3 and talking about dihybrid crosses. Now dihybrid means we're going to be looking at two different traits. We're going to be looking at pea plants today. We're going to be talking about the pea shape, whether it's round or wrinkled, and pea color, whether it's yellow or it's green. So when we talk about dihybrid crosses, we need to remember independent assortment. It's that different characters are inherited independently of each other. So just because I have blonde hair does not mean I'll have blue eyes. So dihybrid crosses look at two characters, and we're going to look at seed color and seed shape today. Now true breeding, we can have homozygous dominant or homozygous recessive. Well, we know that round is dominant and yellow is dominant. And so round will be big R for homozygous dominant, and big Y for homozygous dominant for the seed color. Now the recessives are going to be little r, and little y, and that'll give us wrinkled and green. The hybrids, or the heterozygous, will give us big R and big Y, and that will still give us round and yellow, but it is heterozygous. So, when we cross two heterozygous parents, we have to do a few different things. First off, we take one parent and put it across the top, and we take one parent and put it across the other side. We know that. But how do we get the genotypes, or the alleles, to put on the outside of the boxes? Well, we're going to use a secret trick called FOIL. So you're going to take one parent, and you're going to look at the S's and the Y's, and you're going to use first, outer, inner, last. Let's see how that works. So first would be big S, big Y. Outer, big S, little y, which goes right there. Inner, the little s and the big Y, right there. And the last. That's little s, little y, and that goes right here. That makes sure that we hit all the different combinations. We do the same thing for the other side. We go first, outer, inner, and last. Well, then you know how to do the Punnett square. You bring them down and you bring them across. Now make sure that the S's stay together in this certain instance and the Y's stay together. And if one is dominant, we're going to make sure to put that one first in each grouping. So, if we look at a cross between homozygous dominant and homozygous recessive, now these are really easy to figure out how to put things across the top. There's only one choice. You give a big R and you give a big Y. You got big R, big Y, big R, big Y, big R, big Y, big R, big Y. And for the homozygous recessive, all you can give is a little r and a little y. That goes all the way across the bottom. And then you bring them down and across. Now I'm going to have you do one. We've got heterozygous crossed with heterozygous. So we're going to take one of these and put them along the side, and one of these and put them along the top. And we're going to use that FOIL method. That first, outer, inner, last. So I'm going to give you a second to see how you do. So go ahead and pause and complete your Punnett square. And how did you do? So make sure you check yourself and you write this down. Now I need you to take another minute and actually go ahead and complete your Punnett square by bringing them down and bringing them across. Let's see how you did. Now, you've got the Punnett square done. That's half the battle. The other half is going to be giving us the genotypes with the ratio and the phenotypes with the ratio. You really want to use ratios now because it's not a nice number with 16 boxes to give percents. So you want to give us a ratio, and again, that's just counting the numbers of how many there are. So let's take a look at this. So in this Punnett square, we're going to list all the different genotypes. Oh, we see a big R, big Y. We see a big R, big Y, little y. We see a big R, big Y, and we see a big R, big Y, little r. Little y. We see a big R, little y, and you list them all. And you count them out. Well, I'll tell you a secret. If you can memorize this pattern, the 1 to 2 to 1 to 2 to 4 to 2 to 1 to 2 to 1 ratio, then you'll never have to do a Punnett square for hybrid or heterozygous crosses again. Because all you have to do is see that they're heterozygous and heterozygous, list out the genotypes in this method with keeping big R, big R, big R, big R, big R, big R, and then going through the different combos, big Y, big Y, big Y, little y, little y, little y, and then switch to the heterozygous R's and go through big Y, big Y, big Y, little y, little y, little y, and then go to the recessive R's and go through that same pattern, the big Y, big Y, big Y, little y, little y, little y, and that'll come up with a 1 to 2 to 1 to 2 to 4 to 2 to 1 to 2 to 1 ratio. Now, you need to make sure that all these numbers add up to 16, because there's 16 boxes. If it doesn't, then you've either left out one or you've added one too many, so you need to go back and check. For the phenotypes, you take all the genotypes and tell me what they mean, and then you have to give a total. Because even though that this one means round and yellow, the next one also means round and yellow. And so does this one right here, round and yellow. And so does this one, round and yellow. So all in total, round and yellow, I have 9. And round and green, I have 3. Wrinkled and yellow, I have 3. And wrinkled and green, 1. Now again, there's another trick to this one too. If you can remember that again, you're looking at heterozygous crossed with heterozygous. The phenotypes will be a 9 to 3 to 3 to 1. 9 to 3 and 3 to 1. That's going to be dominant and dominant, dominant and recessive, recessive and dominant, and recessive and recessive. For these two middle ones, it doesn't matter if you put the recessive here first and the dominant on the other side, or vice versa, because both of them are going to be 3s. But one's dominant and one's recessive. Let's try one where there's not an easy trick, that we actually do have to go through and count. So we have big R, big Y, big Y. Big R, little y, little y. Go ahead and do FOIL and create your Punnett square. FOIL's first, outer, inner, last. Take a moment and go ahead and complete this cross. Let's see how you did. So, we've got our genotypes up top, our alleles, and I need you to bring it down, and then I need you to bring them across. How'd you do? Well, let's go ahead and look at the genotypes and phenotypes. So, when we look through, we have a big R, and our big Y, little y. That's one genotype that we have. And we see that we have 1, 2, 3, 4 of those. Then we have a big R, big Y, little y. And we see 1, 2, 3, 4, 5, 6, 7, 8. So we have 8 of those. Then we have a little r, big Y, little r. And that's again going to be 4. Does it add up to 16? Yes, it does. So we've hit all the boxes. For the phenotypes, we're going to tell them what they mean and come up with totals. Well, big R, big Y, little y means round and yellow. And big R, big Y, little y also means round and yellow. And that's going to be 4 and 8, so a total of 12 round and yellow. Then we have our little r, big Y, little y, which means wrinkled and yellow, and we have 4 of those. Does it add up to 16? Yes, it does. And that's it. That's all you have to do. We'll practice this again once we get back to class. I hope you have a great day. Bye for now.